Okay, students, we are going to study MGG staining. Let's know what is MGG stain, what are the principle, what are the procedure. Introduction to MGG staining. MGG stand for May Grunwald Giemsa stain, and MGG stain is a type of Romanowski stain. What is Romanowski stain? So it is a type of polychromatic stain where more than one dye are used, and it consists of both acidic and basic dye. Here we will see that MGG stain is a combination of two stains. One is a May Grunwald stain and other is a Giemsa stain. So because of that we say May Grunwald Giemsa. Okay, and the application for this staining is this is used for staining bone marrow smear, blood smear, and other clinical cytological specimens. When we do the cytological studies, so the cells can also be stained by MGG stain. The principle of this test is used for morphological inspection. That if you want to see the morphology of the cells it is smaller or larger, or if you want to see the nuclear properties, nuclear size, if you want to see the nuclear is to cytoplasmic ratio, so that morphological inspection we can do by doing this procedure. And we can also do differential counting of the cells in the specimen, like if you want to count differential leukocyte count, or if you want to count the cells of different types in the specimen, so we can use this MGG stain. MGG contains alkaline methylene blue, which is a basic dye, and acidic eosin, which is an acidic dye. So there are two combination of the dyes present in the MGG stain: methylene blue, which is a basic dye, and eosin, which is an acidic dye. Stains all cellular component. What does it mean? That this MGG stain usually color the cellular component, different cellular component. Okay, so if you want to see the different cells present in the cells, like nucleus, cytoplasm, so different compartment of the cell will be colored differently by using this MGG stain. Let's know the principle of this test. The basic dyes carry net positive charges, and consequently they stain the nuclei, granules of basophil, and RNA molecules of the cytoplasm. So these are the the component of the cell which are stained by the basic dye, that is a methylene blue. They have a positive charge on it, and because of that it always attract the negative charged component of the cells. So nuclei have a negative charge, granules of basophil have a negative charge, and RNA molecules present in the cytoplasm also have a negative charge. So because of this negative charge component of the cell, the basic dye being positive charge, both have affinity to attract each other, and hence basic dye can give color to the nucleus, it gives color to the granules of basophil, and it can give the color of RNA molecules present in the cytoplasm. The eosin carries net negative charge, and because of this net negative charge, it can attract or it can bind uh, to the positively charged component present in the cell. So it can stain red blood cells, which are positively charged molecules. It can also stain granules of eosinophil, which is a granulocyte. The pH is a very critical factor in staining. So the pH, you have to maintain the pH And if there is any change in the pH, it will lead to wrong staining reaction. So you have to ensure that the stain is properly prepared, and it should have proper uh, pH that should be maintained, so that the proper reaction takes place, so that the basic dye can stain the acidic part of the cells, and the acidic dye will stain the basic part of the cells. The most suitable pH for this stain is 6.5 to 6.8. So the pH is adjusted tightly between 6.5 to 6.8. I hope you are able to get my voice. Is my voice audible?
Yes, sir.
Okay. So we have studied the principle of the MGG staining. Now let's know what are the requirements. Yes, we require sample, we require glass slide, spreader, we require make room wall dye, we require absolute methanol, Giemsa dye, we require sometimes glycerol, phosphate buffer of PL 6.8. So these are the small requirements. In addition to that, we need to arrange microscope so that we can observe the cells. Okay, so there are some other requirements maybe needed, like tissue paper, which is not included here, but this is required. Now, the instruction for preparation of May Grunwald stain. So, how to prepare May Grunwald stain? So, we have to dissolve 0.3 gram of May Grunwald dye in 100 ml of absolute methanol in a 250 ml of conical flask. So, first of all, you have to take a clean, dried 250 ml conical flask, and after that, you have to take 0.3 gram of May Grunwald dye, and you have to 800 ml of absolute methanol. Then after that, you need to dissolve this dye into this methanol. So this my other is willing to join. So we are learning the instruction to prepare the May Grunwald stain. So this is the first step, that we should take 0.3 gram of this dye, May Grunwald dye, in 100 ml of absolute methanol. Warm the mixture to 50 degrees Celsius in a water bath for a few hours, and allow it to cool to room temperature. So this steps is done so that the proper dissolving of this dye takes place in the absolute methanol. Okay, so this is for dissolving the dye, and after that we need to stir the mixture on a magnetic stirrer. So this is another next step is done so that the proper mixing can be done. So we can put on the stirrer, magnetic stirrer, and the dye can dissolve properly in the methanol. We have to leave it stirring for 24 hours. So this much stirring is required so that the complete mixing of the dye with the methanol can be done. And after that, we need to do the filtration process. We have to filter the mixer, and then the filtrate is now ready for use. So this is the instruction we have learned, that how to prepare May Grunwald stain. Next, we have to prepare Giemsa stain. So here is the instruction to prepare Giemsa stain. Take 1 gram of Giemsa dye into 66 ml of glycerol, and warm the mixture in a conical flask for 1 to 2 hours at 50 degrees Celsius. This is the instruction. What we require is, we require a conical flask, and in that conical flask we need to add 1 gram of Giemsa dye and 66 ml of glycerol. And after mixing, we have to put this mixture in maybe in a water bath for 1 to 2 hours. The temperature should be set at 50 degrees Celsius. Cool the mixture to room temperature and add 66 ml of absolute methanol. So this is the another step. that after 1 to 2 hours, we need to cool the mixer and you need to bring to the room temperature. And then after you can add absolute methanol 66 ml. Leave the mixer to dissolve over 2 to 3 days, mixing it at intervals. So here uh, we need to put this mixer for at least for 2 to 3 days so that the dye can dissolve properly. And in that period of time, you need to do mixing uh, at that particular some intervals. Let's say maybe after 5 or 6 hours intervals, you need to mix it properly so that the dye can dissolve properly with the glycerol and then methanol. The stain is then ready for use after filtering. So before using, you have to filter the stain. So here we have studied some instruction how to prepare Giemsa stain and make Bengal stain. Okay. Now the staining procedure. How can we do the staining? So first of all, prepare the smear. Let's say you have bone marrow smear. So prepare the smear, then fix the bone marrow smear in the absolute methanol for 10 to 15 minutes. So we can do the fixing of this smear using methanol, and we should do fixing for 10 to 15 minutes. Prepare an equal volume of make wall stain and phosphate buffer having pH 6.8. So this is needed that you need to prepare the equal volume of this make Grunwald stain and phosphate buffer. Why we are preparing the mixture of this phosphate buffer and make Grunwald stain? The purpose is that we can, when we use the phosphate buffer of 6.8, so the buffer, the stain pH can be maintained within 6.8, so that the proper staining can be done, proper staining reaction takes place. Okay, so then mix well when we prepare the mixer. So we need to mix well properly, and after that we have to pour the mixer on the slides that contain spear, and you have to pour so that there can be the smear should be covered properly. Okay, so slides to fully flood on the slides. Okay, and stain for 10 minutes. So completely you have to flood the mixer over the sphere on the slide, and just wait for 10 minutes, right? So this is the time you have to provide so that the proper staining will occurs. Next, prepare 1:10 dilution of Giemsa stain with the phosphate buffer with pH 6.8. Then mix well. This is what we need to take 1 part of Giemsa stain and 9 part of phosphate buffer, so it becomes 1:10. And this dilution mixer you have to prepare during this staining period 10 minutes. Okay, so once you have prepared Giemsa stain, diluted Giemsa stain, then we have to mix well, and after 10 minutes of May Grunwald staining, when the time for this May Grunwald staining is over, then now we have to pour away the May Grunwald stain of the slides. So you have to just remove the content of the May Grunwald from the slides. Okay, just pour away means you have to remove the May Grunwald stain of the slide from the slide, and you can do the next step that you have to pour the Giemsa mixer now. The diluted Giemsa mixer now you have to put on the slides, and you have to wait for another 15 minutes. This is very simple procedure. That first of all you have to stain with the May Grunwald stain for 10 minutes, and the next you have to stain with the Giemsa mixer or diluted Giemsa stain for another 15 minutes. After 15 minutes, pour off the stain means remove the stain, and you need to flush the slides with the running tap water. That should be done gently to take care of the removing of or destruction of the smear. So do not harm the smear. Just when we put the smear under the tap water, running tap water, so just ensure that the tap water do not wash away the smear. Okay, so next is a clean excess stain with the wipes. So there is a tissue paper or wipes are there. That wipes can be used to clean the excess stain from the back side of the slides. Now you can do the air drying, or you can use hair dryer for 10 seconds. Okay, no longer than 10 seconds. So hair dryer can also be used for at least for 10 seconds. And this is for drying is done. So when staining is over, you should do drying process. So hair drying, or if you want to speed up the drying, you can use the hair dryer also. Next step is to place the long cover sleeve on the area of the interest. Means the smear over you can put on the smear, you can cover the cover sleeve, and after that you can examine under the microscope. So this cover slip is usually kept to prevent you know uh, you can prevent attachment of dust particles to the smear. You can also use DPX so that you can prepare the permanent slide. So first of all, you can put the DPX, and then you can put the cover slip, and the slide become permanent. And when it dry, then you can now examine it at the microscope under foil immersion objective. So this is what the procedure we have studied. There are different steps are there, and the procedure is very easy. First of all, you have to stay in with the milk renewal stain for 10 minutes. Then you have to wash off the stain. Then again you pour with another Giemsa stain, which is diluted one, for 15 minutes. And after 15 minutes, you can wash out the stain, and you can put the slide under the running tap water, so that you can remove the excess stain which is stick with the smear. And using the wipes, means tissue paper or like a tissue cloth, so you can clean excess stain if it's stuck with the slides from the back side or from the front sides. And then after that we can do the air drying. And if for air drying it takes a longer time, so you can also use hair dryer for not longer than 10 seconds. And once it is dried, we can place a cover sleeve over the smear area where you want to observe the cells. And now the slide is now ready for the examination. So this is what the observation. You can see the one smear at 400 magnification, means at 40x. This is what the view is observed. Now we can change to the 100x while immersion objective, and you can be able to see the details of the cells, each and every cells. So this is a 400 magnification times where the bone marrow cells can be seen, where you can be able to trace out the mature cell, blood cells, okay, and erythropoietic cells, lipopoietic cells of different origin. So bone marrow smears can also be, stained, or blood smear can also be stained. Psychological specimen can also be stained with the MGG stain. If you do the blood smear, and if you make a blood smear, if you want to stain with the MGG stain, so you can see the interpretation of the result, that it look like erythrocyte can be seen as a light pink to brown color. Lymphocyte can be seen from blue to purple nucleus, and eosinophil, there can be the blue violet color. Eosinophilic granules can appear as a deep red color, and neutrophil it appear as a deep blue violet. Neutrophil granules appear red color, and basophil blue violet. So this is what we have discussed about the MGG stain, the principal procedures, and instruction to prepare make renewal stain, Giemsa stain. Now we can discuss if you have any doubt.